I think we'll get started. I just want to welcome everybody. I see we have a nice large crowd. And this is LEV Becker's promotion talk. And just a little history about Lev. He received his bachelor's in science at Queen's University in Kingston, Ontario in 1997, and then went on to get his PhD at Queen's University with Marlis Koshinsky. And then in 2004, moved to Seattle to do a postdoctoral fellowship with Jay Haneke at the University of Washington. And following that, he came to the University of Chicago in the Department of Pediatrics as an assistant professor, where he stayed until 2015, at which point he shifted over to the Bentley Department for Cancer Research, which is where he is right now. And Lev has had a recent history of working on macrophages in health and development and disease, both in atherosclerosis, diabetes, and now cancer, and we'll hear some of that today, I'm pretty sure. I did also want to mention that he's received several honors along the way, including the J. Clifford Moose Endowment Award for Cancer Research, and also the Cancer Research Foundation Investigator Award. So it's my pleasure to introduce and welcome Lev, and there's a couple more seats up here for anybody who wants it, and otherwise I guess we'll get started. You can read the title of this talk in the Community Cell Platform for Identifying Disease-Producing Mechanisms and Developing Novel Therapeutics. And also, welcome.
Yeah, thanks Jeff, and thanks everyone for coming. So I'm going to take you through some studies that we've been doing in my lab for the last 7 years. Our work really started with macrophage biology, really focused in diabetes, but we've extended this and really turned this research around macrophages and now other innate immune cells. We try to create a platform where we can study their role across a broad spectrum of disease. And I'll show you some of the projects we've been working on. So basically, although the disease that we're studying varies, we always take a very similar approach. And most projects in the lab, combine studies in humans. We really like to start here so that we can ensure that the pathways that we're studying are clinically relevant. Can then go back into the mice to test, to use the power of mouse genetics to test physiological mechanisms, and then ultimately, of course, to cells. To identify the signaling components within cells and the functional changes that are driving the phenotypes. And we have a broad spectrum of approaches that we use across all the projects in the lab. But I'm going to highlight these two because we often use proteomics analysis of disease versus control, preferably from humans, as our starting point to ask what's wrong, what's different in these cells from patients with disease to control. And then can we understand how those differences are regulated and how do they contribute to disease? And so as Jeff mentioned, we're studying these innate immune cells in models of infection, cancer, atherosclerosis, diabetes. And there's a lot of crosstalk between these diseases, and I'll show you some data to that effect. And obviously, studying a wide range of diseases means a lot of red tape is required. And we've certainly paid our fair share of dues in that respect over the last 7 years. And so the first project I'm going to talk to you about is studying the role of macrophages in obesity, insulin resistance, and type 2 diabetes. And this work was done by my first graduate student, Brittany Coates, who's now moved on, as well as Kelly Schoenfeld, which was the first technician that I hired when I came to University of Chicago, and our clinical collaborator at University of Washington. And what you're going to see in this series of data is, one of the key measurements that we made was to compare macrophages from this diabetic state to the infection state. And this comparison really moved this project forward. And so if you take a step back, it's really hard to read a macrophage paper without encountering this M1 and M2 subclasses of these cells. And this has been classically how macrophages were defined. They were defined based on infection paradigms, where these M1 type macrophages play a pro-immunogenic, pro-inflammatory function that promotes tissue destruction as well as protects us from invading organisms, while these M2 type macrophages promote tolerogenic and anti-inflammatory responses and mediate repair. And this M1 state has really been defined in infection. And what has happened over the last 2 decades is we've taken this understanding of the pathways that are altered during infection and started to understand whether they might contribute to what I'm going to refer to as sterile inflammatory conditions. So these are conditions of chronic inflammation, but that are not actually driven by an infectious organism. And this strategy has been supported most heavily in obesity and type 2 diabetes, where we can knock out components, signaling components that are driven by bacteria and see phenotypes in metabolic disease. And there's been probably hundreds of papers on this that have produced this paradigm where in the lean state, adipose tissue is small and you have these protective M2-like macrophages, while during obesity, adipose tissue expands and you get the recruitment of these M1-like macrophages, which then promote insulin resistance during type 2 diabetes. And while this work was supported by numerous studies, one of the challenges is if you go look for a bona fide M1 macrophage in humans and mice, no one has been able to find one. And so roughly, I guess, 4 years ago, we found that they're not there because they don't exist. And we defined a new type of macrophage, which we called metabolically activated, or MME. And this phenotype was being driven by saturated free fatty acids, which for a long time were thought to mimic LPS action on a macrophage, linking infection to metabolic disease. But what you can see from this graph here, which is a plasma membrane proteomic study of MME, M1 versus control, and each dot is a protein, you can see the effect of bacteria on a macrophage is changing many proteins, as is the effect of fatty acids and metabolic dysfunction, but that they are completely orthogonal to one another. So these cells don't look like each other at all, outside of the fact that they're both overexpressing cytokines. Okay, we then used this proteomics approach to develop biomarkers that we could then look for in patients and in mice. And I won't take you through all of the data, but here are some MME markers that we found. And you can see in obese patients, you see very strong and uniform expression for these MME markers on the cell surface. And in contrast, if we went into patients with cystic fibrosis, this is a chronic bacterial infection that takes place in the lung, resulting in airway damage and very high morbidity. These macrophages looked just like we would predict. They are expressing the M1 markers, which are reflective of exposure to bacteria. Okay? And then we further went on and showed that the expression of these MME markers on macrophages in adipose tissue were very highly correlated with BMI, which is a measure of obesity in patients, and of course the M1 markers, which were not expressed to any significant extent, were not. In addition to identifying markers that we could use to track these populations in humans, the plasma membrane proteomics also gave us insights into the mechanism driving these two distinct phenotypes. And we used two approaches. First is by looking at which proteins were differentially expressed in each state, we can use informatics to work backwards to ask what pathways might have produced those changes. And secondly, and this was a little bit of a surprising finding, we found that signaling proteins were actually being identified in our plasma membrane proteomics analysis, likely because they were being tethered to the plasma membrane proteins through protein-protein interactions. And so if we looked at the three different cell types here, M0 is the control, the metabolically activated, the classically activated, you could see, for example, STAT1 is preferentially recruited to the membrane in the M1 macrophages, And consistent with this, informatics analysis showed that most of these proteins were driven by the type 1 interferon response. In sharp contrast, in the MME state, we saw P62 to be recruited to the membrane, and informatics analysis suggested that PPAR gamma regulated proteins were the ones that were overexpressed on the cell surface. Now, I won't take you through the data, but we validated using knockouts and inhibitors that these two pathways were disparately regulating these two states, and came up with this final summary diagram where in adipose tissue, free fatty acids like palmitate, which are released by insulin-resistant adipocytes, are causing this MME phenotype by binding to cell surface receptors to drive NF-kappa B, but also getting internalized by the cells to induce P62 and PPAR gamma, which are controlling these cell surface proteins which are involved in lipid metabolism. And that makes sense. The cells get lipid inside them. They then want to express proteins that are going to help them to handle that lipid. On the other hand, LPS, it binds to its TLR, turning on NF-kappa B, and the cell surface proteins that we found were actually being induced by the type 1 interferon response. And so this is a very specific description of what's happening in these two states, but the more general message is that the pathways for how we detect inflammation, in other words, NF-kappa B, which is giving cytokine production here, is distinct from the mechanisms that are supporting cell surface protein expression. And in this way, you can have two cells that have inflammatory cytokine overproduction manifesting in two completely different cell surface proteomes, making it very challenging to take markers you identify in one disease state and translate it to another. So having found this new macrophage, the next question we wanted to ask is, what are these cells, how do they contribute to obesity? And there's a series of papers that showed that saturated fatty acids, such as palmitate, are known to increase ROS production in macrophages. And there was another paper from Ira Tabis's group in Colombia, which showed that TLR2 and NOX2 mediates the signaling from oxidized lipoproteins on macrophages. And so we thought that maybe fatty acids might act similarly to these oxidized lipoproteins and be dependent on TLR2 and NOX2 for their signaling. And that was indeed what we found. So this, on the left panel here, you see wild-type macrophages with IL-1 beta being induced or IL-6 beta being induced. And then if we knock out NOX2, this is the GP91 subunit, or TLR2, we can attenuate inflammatory cytokine production. And importantly, these knockouts don't at all interfere with inflammatory cytokine production from bacterially challenged macrophages. So this is saying that even the way in which NF-kappa B is being activated in these two systems is distinct. And we could validate this in vivo. So we could take mice, I'll focus on the red bars first, put them on a high-fat diet, and you can see TNF, IL-1, and IL-6 are all elevated. And then if we do a myeloid-specific GP91 knockout, we can attenuate inflammatory cytokine production right in the adipose tissue from the mice. Now, as I mentioned, the ability of these macrophages to make cytokines is what drives insulin resistance during obesity. And sure enough, if we put mice on a high-fat diet for 8 weeks, we observed protection in terms of glucose tolerance in these macrophages that lack NOX2, or in the mice that lack NOX2 in their macrophages. But what was really surprising was that if we put these mice on a high-fat diet for longer, this protective effect went away, and we actually even needed more insulin to maintain this glucose tolerance, suggesting the mice were becoming insulin resistant. And we could test that using an insulin tolerance assay. The take-home message here is that the mice were indeed severely insulin resistant. And so this kind of posed a really important question to us, is how can the same genetic knockout produce a protective effect early on and produce a worsening later on? And to begin to answer that question, we went back to our proteomics approach. We took murine MME macrophages and asked what proteins on the surface were elevated and which ones were suppressed. We then did bioinformatics analysis of these proteins and found the ones that were induced were obviously enriched in plasma membrane, membrane raft, matrix. That told us we did a good job of isolating plasma membrane proteins. But the surprising result was the appearance of these lysosomal membrane proteins on the cell surface of the macrophage. And this was very specific to the MME phenotype, because if you challenge MACs with bacteria, you don't see this. Now, lysosomes appear on the cell surface of cells when lysosomes are exocytosed. The membranes fuse, and you see lysosomal membranes proteins on the cell surface. And we validated that using multiple approaches. And importantly, we also see that in vivo. So if you look at MACs from a low-fat diet versus a high-fat diet, we see many of the same lysosomal membrane proteins on the cell surface. So you might be asking yourself, well, what does lysosomal exocytosis have to do with obesity? And when I was visiting to give a talk at Weill Cornell, I met a colleague by the name of Fred Maxfield who studied this extensively. And one of the things that macrophages do in adipose tissue is they clear dead adipocytes, okay? And I've tried to draw this to scale. No matter what a reviewer of one of our papers says, this cell cannot eat that cell by phagocytosis, okay? So instead, what it does is it exocytoses, it forms fusion with the cell surface of the adipocyte and exocytoses lysosomal acid lipase, which then takes this triglyceride, which makes up most of the adipocyte, turns it into fatty acids, and then those fatty acids get internalized by the MAC. Now, even if you're exocytosing, that's a big meal for one MAC. And so he calls his friends and her friends. And you can actually see the appearance of these crown-like structures in vivo with independent nuclei. The green is the macrophage stain surrounding this perilipin negative dead adipocyte. And so we thought that perhaps one way in which there could be worsening is that if we interfered with this process. And so Fred had this wonderful assay to monitor lysosomal exocytosis to dead adipocytes in vitro. And what's happening here is you take the macrophage and you label it with FITC dextran biotin beads. Those beads end up in the lysosome, and then you incubate it with avidin labeled live or dead adipocytes. And so when the macrophages exocytose their lysosomes, that fluorescence would then be captured on the cell surface of the dead adipocyte. And so if you just take a look at the wild type, you can see that when you incubate MACs with dead adipocytes, you see this really nice fluorescence captured on the surface of the adipocytes, but that doesn't happen to a large extent in live. And importantly, when you knock out NOX2, you effectively block the ability of these macrophages to exocytose their lysosomes. And this doesn't just happen in vitro. If you isolate macrophages from obese fat, you can see those cell surface lysosomal proteins are all attenuated in vivo as well. And so if macrophages can't clear dead adipocytes, what happens? Well, the fat becomes fibrotic, okay? And you see the accumulation of massive amounts of these crown-like structures because the macrophages aren't able to clear the dead cells and let new cells come in to absorb new nutrients, okay? And when fat can't store nutrients, it usually goes to the liver. And that's exactly what we saw in these mice on the high-fat diet, is a tremendous amount of hepatosteatosis. And this is what is driving the insulin resistance in the late phase. And so this led to this final diagram here where fatty acids released by adipocytes early on are supporting inflammatory cytokine production as well as exocytosis. And these cytokines can promote insulin resistance, but this exocytosis can protect against it by clearing dead adipocytes and sparing the liver of the fat. And so you might ask yourself, why is there a time delay between these two processes? And it's very analogous to what you see in a tumor. So early on, adipose tissue is small, and as it expands, angiogenesis can keep up with the expansion of the tissue. But once it reaches a critical mass, angiogenesis can no longer keep up, and you start seeing hypoxia as measured here by HIF at 16 weeks on the high-fat diet, where we see the worsening, no effects at 8 weeks. And we can also show that the adipocytes are indeed dying by a caspase-3 activity assay later on, but not early on. Okay? And so I'm just going to kind of summarize the first section for you according to this model and the sequence of events. So basically, we found this new phenotype by comparing humans, obese humans with patients with cystic fibrosis. We then used our proteomic strategies to identify mechanisms, then went back into mice to understand what functions they're playing, and identify a whole new suite of ideas that we hadn't known before. And now we're going back into the humans to ask, is this lysosomal exocytosis and linked to hepatosteatosis also observed in patients? And that's all been going on in the background. We've had a parallel study in humans where we're comparing lean to obese insulin resistant to obese insulin sensitive. And the original structure was just to look at visceral and sub-Q fat and do immune profiling, macrophage proteomics, adipocyte sizing, and correlate that to parameters. But based on our findings that were happening at the same time, we were dynamically able to alter this in a way where now we're going to focus our proteomics to look at these lysosomal proteins. We're going to measure adipocyte death using these two approaches. And we've also now started to do liver MRI scans on all the patients so we can actually quantify the extent of lipid accumulation in the liver. So this is just a wonderful example of how this back and forth approach between humans and mice can drive ideas in the mice that we can find and then go right back into the humans and ask whether those ideas are clinically relevant. And just to give you a little update, I think we're up to maybe, our goal was to get around roughly 50 patients. I think we're up to about 35 or 40. And I was really happy to see that we are able to find patients that are equally obese but differ in insulin resistance. And this is clinically really important because being insulin resistant and obese is much worse than just being obese. Okay? And so one of the other projects that came off from this MME macrophage discovery was a collaborative project between my lab and Marcia Rossner's lab. It was really performed by Payal Tiwari, a graduate student who was co-mentored by the both of us, and Arian Blank, who's an undergraduate student who's worked on this project for 3 years now, I think, or maybe even longer. I don't have time to give you all the details, but I'll take you through a quick summary. And so basically the idea here is that obesity is a really strong risk factor for triple-negative breast cancer, a type of breast cancer that has very poor outcomes and few therapeutic options. And our thought process going in was that perhaps local inflammatory cytokine production driven by macrophages in obese mammary fat might be promoting tumor stem-like properties and promoting tumor initiation and growth. And so, like I said, I'll just give you the very quick highlights here because I have other work I want to discuss as well. But we were able to get some human samples from Seema Khan at Northwestern. And what we were able to show here is that just like I showed you earlier in visceral fat, if you isolate macrophages from mammary fat of obese women, you see an increase in the macrophage number, and that MME phenotype is what's causing this. That M1 phenotype is really pretty much absent in the mammary fat. This is important because M1 macrophages are thought to oppose cancer development, not promote it. But the role of MME had not been found. And so what Payal showed is that if you take these macrophages either from humans or from mice or from in vitro, in vivo, they do indeed induce a stem-like phenotype based on marker expression. She also showed various functions that just as short as 24-hour exposure to these macrophages make cancer cells adopt this stem-like phenotype and can promote invasiveness as well as tumor initiation. And we then went back into the mice. And so one approach to looking at stem-like properties in mice is to do what's called a limiting dilution assay. and determine whether tumor incidence can be supported with a much smaller number of cells. And so what Pyle showed here was that if you take mice and put them on a high-fat diet for 12 weeks, you can start seeing tumors that are initiating at doses of cells that you cannot get tumors to format in lean mice. And she showed it with one TNBC cell line with a second TNBC cell line. And then using that same myeloid cell-specific NOX2 knockout, she was able to show that this effect could be lessened when macrophages lacked NOX2 and were incapable of becoming metabolically activated in the mice. And so this paper just got submitted, and so I hope that it'll come in press soon. So I'm going to shift gears a little bit here to talk about another project, and this is going to be talking about how obesity and diabetes promotes atherosclerosis, and there's going to be some important implications to infection as well in this story. So the statistics are actually pretty staggering here. 9% of Americans have type 2 diabetes, 30% of them are pre-diabetic and are going to become diabetic. And people with diabetes don't die from diabetes for the most part. They really die from the large majority of them die from cardiovascular disease. And of course, cancer is the other major part of morbidity. And so CHD risk is actually increased by about 4 times. And it's said that the probability of a patient with type 2 diabetes developing a myocardial infarction is as high as a non-diabetic who's already had one infarction to have a second one. So this is a massive clinical problem. And anti-diabetic therapies are not treating it. You can lower the diabetes, but you're not going to lower your cardiovascular risk for the most part. And so we had a very simple hypothesis. There's a large amount of data saying that type 2 diabetes inappropriately activates macrophages. And there's even larger body of evidence suggesting that macrophages play a central role in atherosclerosis. They accumulate cholesterol to make foam cells. And so what you're seeing here in this cross-section of the artery is this lesion, and a large majority of this lesion is macrophages that have eaten cholesterol. So atherosclerosis can really be thought of as a macrophage disease. Obviously other cells contribute, but this is one of the major ones. And so the idea was very simple. Diabetes alters the MAC in a way to promote atherogenesis. Although it's simple, it's actually been very difficult to test experimentally. And the reason is that type 2 diabetic patients, they are often eating high-fat diets, and their plasma cholesterol levels are also elevated. And so since cholesterol is a major risk factor for atherosclerosis, it's been challenging to dissociate these effects. Of course, in humans, one can use statistical approaches to show that diabetes risk is independent of this cholesterol. But the challenge is in mice, where these are atherosclerotic-prone mice, if you feed them a low-fat controlled diet, a diet that induces hypercholesterolemia only, or a diet that induces hypercholesterolemia and diabetes, you certainly see body weight's elevated, glucose is elevated, insulin's elevated, so you see the diabetic phenotype, but here's the problem. You also see an elevation in the plasma cholesterol. And so how do you dissociate cholesterol-mediated events from the diabetes-mediated events? And so our strategy was a fairly complicated one, but essentially what we did is we set up models where we could get and isolate each variable. So using wild-type mice, we can make them just have obesity and insulin resistance, or we can modify the diet in these atherosclerotic prone mice to have hypercholesterolemia only or both. And the parameters are shown here, they're very clear. The idea here then was then to isolate macrophages, perform proteomics analysis, and then ask 2 questions. First is obesity and insulin resistance sufficient to induce a pro-atherogenic program in macrophages? And second, is obesity insulin resistance necessary to induce this in the context of hypercholesterolemia? Okay? And then because the cholesterol changed, we then had to identify what the mechanism was first, and then block this mechanism and show that you only get a phenotype when obesity is present, not when it's absent. And I know it's complicated, but that's kind of the overarching idea. I'll take you through some of the data. So we studied the macrophages by proteomics. And again, on the left here is just obesity only, cholesterol and obesity, and just cholesterol. And what you can see is we found a subset of proteins which are beautifully regulated in a similar way when obesity is present, but not when it's absent. And again, this is looking at macrophage cholesterol levels, showing that in the obesity model, there is no cholesterol accumulation in the MAC. So these effects were unlikely to be driven by cholesterol. Why? Because these guys are cholesterol loaded, and that same pattern is not being observed. Okay? When we look at these proteins a little closer, 3 of them, lactoherin, ApoE, and complement C3, are known to be anti-atherogenic. And they're known to be anti-atherogenic because if you knock them out in macrophages specifically, you promote atherosclerosis. Okay? So the simple idea here is obesity is attenuating anti-atherogenic proteins in macrophages. And 2 of these guys, ApoE and C3, are known to be involved in lipid metabolism and cholesterol movement. So one idea is that obesity might, by downregulating these proteins, promote cholesterol accumulation in the macrophage. And that's exactly what we saw. If you give cholesterol to a macrophage from a lean animal, it will certainly accumulate some, but if you isolate that same macrophage from an obese animal, you can have even greater cholesterol accumulation. And we can transfer this defect from lean mice or from obese mice to lean mice by simply knocking out either one of these 2 proteins. They mimic the effect of obesity in terms of promoting cholesterol accumulation in the macrophage. And so I won't take you through how we got here, but we got to the point where we thought interferon gamma might be playing a role. And so if you look at T cell production of interferon gamma, you can see that you need the presence of obesity to induce its expression. You don't see it by hypercholesterolemia alone. And then if you do a myeloid cell specific interferon gamma receptor 1 knockout, you can effectively reverse this pattern in the mice. Whereas when obesity was absent, there was absolutely no effect. So obesity needed to be present for gamma to be induced, eliminate gamma signaling, you reverse this phenotype in the obese mice. And I showed you those 2 key proteins that were promoting foam cell formation or cholesterol accumulation. And just as you'd predict, you see an attenuation of foam cell formation only in the obese mice, not in the lean mice, because this gamma signaling event is not taking place. You could extend that further and quantify lesions. You see an attenuation of lesion progression when receptor 1 is knocked out in the max. In the obese mice, no